My name is Phan. I'm a mentorship student from Dulles High School, and my presentation today is about the microbiome in the context of maternal fetal health. And so before I get into my project in specific terms, I wanted to really give a broad overview of the work that I did in my mentorship program. I was really fortunate to have the opportunity to be mentored at the Agard Lab at Baylor College of Medicine, which is a maternal fetal medicine laboratory that investigates the role of the epigenome and microbiome on human health. So most prominently, their research first proposed an independent fetal microbiome that is unique from maternal environments. And under the Barker hypothesis, modifications to this fetal microbiome can have consequences on adult disease and health. So specifically, I did work on a disease called Coriomenitis, which is an infection that does modify this fetal environment. And so I'm going to take some time to define some terms in my introduction. And I wanted to first refer to the actual first sentence of my abstract, namely Dr. Agard's lab seeks to identify the role of environmental exposures in preterm births, specifically looking at modifications in the human epigenome and microbiome. And so you may understand inheritance in terms of the human genome. You may have heard that DNA carries genetic information and that genetic information is passed from generation to generation. And so epigenetics is the study of modifications to gene function or phenotypes that occur without an actual change in the DNA sequence. So as you can see here, changes to DNA can occur at the genetic level in which a mutation occurs and then the phenotype or resulting function is modified, or it can occur at the epigenetic level in which a marker attaches to the genome and changes the resulting function. And so the microbiome can be defined as a microbial community occupying a reasonably well-defined habitat. And so in the context of human beings, this means the bacteria that occupy and colonize our organ systems. And so, for example, you may have heard of gut bacteria or gastrointestinal bacteria that help us digest. And through the Human Microbiome Project, researchers have actually identified a massive variety of microbes that colonize the human body, actually a greater number of microbes than there even are human cells. And so within humans, there are approximately 5 zones with almost completely unique microbial environments, namely the airway/nose, the mouth, the skin, the gut, and urogenital regions. And so the bacteria that inhabit each of these areas, and even those that inhabit specific organs, have really unique environments in which they can survive, and they each perform really unique and specific functions. And within organ systems, there's a proven interaction on both a structural and microbiotic level, meaning that disruptions in the microbiome can manifest in a variety of diseases. And so going back to the gastrointestinal microbiome, if you've ever taken antibiotics and gotten a stomach ache, you've actually experienced this. Broad spectrum antibiotics can wipe out the really important functional bacteria that aid in many of our body's processes, including digestion. And so Dr. Agard's lab specifically looks at the environment in the uterus, which is aptly named the intrauterine environment for bacterial growth. So now that you have a bit more background, I kind of want to explain why this research is so significant. And so in their paper, The Placenta Harbors a Unique Microbiome, Dr. Agard's team established that the placenta and thus the entire environment around the fetus may actually contain colonizing bacteria, whereas previous research thought it to be sterile. Furthermore, her research into the vaginal microbiome has established a mismatch between the mother's vaginal microbiome and the baby's gut microbiome, which indicates that this microbiome does not simply come from microbial exposure during vaginal birth. And just to elaborate a bit, the bacteria in an infant's first stool, which is also known as the meconium, does not match the bacteria in the mother's vagina, which kind of indicates that the bacteria comes from somewhere or develops from somewhere within the in utero environment and not from exposures during or after birth. And so this can be understood within the context of the developmental origins of adult disease hypothesis, which is also known as the Barker hypothesis, or the idea that adverse fetal disturbances can result in adult diseases. And so as rates of metabolic diseases are higher than ever before, this really explains how diseases such as obesity and diabetes can be passed from generation to generation, as well as how certain prenatal environmental exposures can manifest in later metabolic disease. And so Dr. Agard's work in the placental, pregnancy, and newborn microbiome has really completely transformed our understanding of microbial development. And by looking at interactions between these cellular communities and their environment, this research provides insight into the connection between irritants, pollutants, and common adult diseases. Finally, I want to introduce how Dr. Agard and other researchers study modifications in these environments, namely the concept of bioinformatics analysis. So bioinformatics uses computation to better understand biology. The model of a protein on the right is a really fantastic example of how this kind of technology can really help us understand and model biological concepts. And so I'll be discussing some types of bioinformatics analysis later, but I really just wanted to introduce the idea of using computer science to understand all these levels of genetic composition, epigenetic markers, and composition in microbial communities. And so Dr. Agard's lab does investigate a variety of pollutants and infectious diseases, but the principal infection that we will be discussing today is chorioamnionitis. It's also known as triple I, which stands for intrauterine inflammation and or infection. It is a polymicrobial, non-traditional infectious disease. And so to understand chorioamnionitis, we must kind of understand the fetal intrauterine environment. So placental membranes as a term are often used to describe all the fetal components of the placenta that we see here. But relevant to chorioamnionitis, the key membrane that surrounds the fetus during pregnancy is the chorion and the amnion, which are the two layers of that membrane. And so historically, chorioamnionitis is thought to be an ascending infection. So some kind of bacteria in the urogenital region would spread up through the cervix and into the fetal environment. And so risk factors for chorioamnionitis and similar infections have historically included prolonged duration of labor or premature rupture of these membranes. Chorioamnionitis is also known to be linked to this genus of bacteria, Ureaplasma, of which there are two bacterial species that colonize the human body, U. parvum and U. ureolacticillum. And so I wanted to also talk about why chorioamnionitis is so important to study. Firstly, that histological chorioamnionitis is thought to complicate about 40 to 70% of preterm births and 2 to 4% of term pregnancies. So it's a really extremely important issue when addressing the topic of preterm births and maternal fetal health. You may have also remembered that I called chorioamnionitis a non-traditional infection, and this is because of the low virulence and long colonization period associated with the organisms that cause the disease, which leads to a kind of lack of traditional infection symptoms such as fever. In addition, the fetal and neonatal immune system is much weaker than the adult immune system, which results in an overall less active response to infection. And therefore, chorioamnionitis infections can present as asymptomatic until we analyze them retrospectively. However, one of the most significant and devastating manifestations of chorioamnionitis is fetal inflammatory response syndrome, or FIRS, which is a condition that's associated with fetal organ injury. So I also wanted to touch on how we trigger chorioamnionitis for research in the lab. As previously mentioned, the bacterial genus Ureaplasma has been overwhelmingly implicated as correlated with chorioamnionitis infection, as studies dating back to 1975 have identified associations between the abundance of Ureaplasma in the urogenital region and risk of the development of chorioamnionitis in patients. But as it takes time for cells to recognize and respond to bacterial infection, other methods are used to clinically trigger chorioamnionitis as well. The lipopolysaccharide, or LPS, membrane of the Ureaplasma bacteria is one of them. It's a molecule consisting of a lipid and a polysaccharide, which forms the outer membrane of gram-negative bacteria. And so cells can recognize the LPS of these foreign particles and cause an immune response really similar to infection by the bacteria itself. And so chorioamnionitis and many other infections is also associated with elevated levels of certain interleukins, or ILs, which are naturally occurring proteins that mediate cell communication. So in the case of triple I, IL-1 beta can be used to induce a cellular response, and elevated levels of IL-1 have been shown to be critical for the progression of chorioamnionitis. And so this is seen as one of the methods with the shortest time points, as it directly triggers the infection on a cellular level rather than waiting for immune recognition. And so I also wanted to touch on successes in the field and what we know about chorioamnionitis thus far. For obvious reasons, most of this research is done in animal pathological models rather than in human models and human mothers. And so as I discussed previously, chorioamnionitis is associated with elevated IL-1. This helps us kind of understand the pathway for this disease development. One major symptom of chorioamnionitis is this clinical lung maturation. So it decreases the spaces between the alveoli, which are the modules that we see here in the lung. And this leads to rubbing or abrasion of these alveoli. And the scarring can have consequences on the health of the neonate, the infant, et cetera. And another name for LPS is endotoxin, and it's associated with a condition called endotoxin tolerance, in which LPS is no longer efficiently recognized in the body. And this can lead to an innate immune tolerance in the fetus that makes it difficult to recognize and fight future illnesses. Chorioamnionitis has also been associated with alterations in other organs, specifically thymic involution, which just means shrinkage, and responses in the spleen and lymph nodes. And so this category of organs known as lymphoid organs, we're still trying to discover precisely what causes the changes to them. Chorioamnionitis is also associated with a condition called necrotizing enterocolitis, which is an infection of the digestive tract, as well as severe consequences of brain inflammation and injury. And so referring back to the condition of FIRS, which occurs as the result of chorioamnionitis, some scientists theorize that chorioamnionitis is compartmentalized in the body to prevent as much brain damage as possible. So for example, the inflammation of the lung, the skin, the gastrointestinal tract, and the chorioamnion, which are all substances that do touch this infected amniotic fluid, might actually prevent the infection or fight the infection from moving to the brain. However, this doesn't provide an explanation for what mediates this compartmentalization, what causes the changes within the previously mentioned lymphoid organs, and which organs are responsible for signaling. And so there are some treatments available for chorioamnionitis, namely different types of antibiotics and maternal betamethasone, which is a corticosteroid. These treatments are often given to mothers who experience membrane rupture as a preventative measure for chorioamnionitis. And so we have a lot of information for this disease, but also quite a few questions, which kind of leads us to the gap that Dr. Banerjee's research addresses, which is what is the role of the microbiome in all of these processes? And so just one more time, I'm going to give you some background to better understand this research. I created this diagram to represent a generic bioinformatics workflow for the human microbiome. So let's pretend for a minute that we are in the lab and we just collected a sample from the placenta. So really, what do we do next? Firstly, the DNA must be isolated in order to be sequenced. So we burst the cell open or lyse it, and then we remove everything that's not relevant to our research. And then we send the isolated DNA to a sequencing facility. And so there are many different types of sequencing, but two important ones for bioinformatics of the human microbiome are WGS or whole genome sequencing and 16S amplicon sequencing. And so in 16S sequencing, only a really small region, the 16S subunit of ribosomal DNA is actually analyzed. And so amplicon sequencing can only identify taxonomy at a broad level, often not giving enough detail to identify species or strain. Dr. Banerjee actually explained this to me as trying to identify a person by just having their arm or just having their leg. You might be able to tell what kind of person they are, but it's very difficult to identify an individual by just a limb. In contrast, whole genome sequencing cuts the entire chromosome into small overlapping fragments that can be put together like a jigsaw puzzle. And this results in sequence data that can be used to determine the relative abundance and functional potential of organisms. However, because a very large number of sequences for each sample are made with this method, it creates a really complicated process for downstream analysis. And although both methods can be used to compare microbial communities, one might be chosen over another, depending on time constraints, desired level of taxonomic identification, and research intention. Unfortunately, I will not have time today to discuss the methods by which we filter, sort, and cluster this data, but all you really need to know is that we end up with a series of outputs that allow us to analyze and characterize this microbial community and specifically analyze different metrics of bacterial diversity. And so finally, we can take a look at some of the project that I actually assisted on. And so the title was LPS, IL-1 beta, or U parvum-induced chorioamnionitis in primates alters inflammation in the resident microbiome. And so I actually helped design this figure as well. Here we see that 54 rhesus macaque dams were given sterile intra-amniotic injections, so injections into the amniotic fluid of either saline in the control cohort or a specific chorioamnionitis trigger. So I hope these terms are a bit more familiar now. RHIL1, LPS, UP, which is U parvum, and one cohort that was given both RHIL1 and LPS. And so notice that, as I've said, these methods have different time points. And then to collect data, the infant macaques were delivered via cesarean delivery and swabbed in multiple areas relevant to our research. And also notice here that microbiome studies are a tertiary method to these pathological methods here. And so here is the supplemental figure for the study design, something I also assisted with. And here we can see the reappearance of many of these terms, WGS versus 16S sequencing, all of these different treatments, et cetera. And I hope now that you kind of know what they are, it seems a lot less complicated than the acronyms might make them seem. And all these letters really just represent different treatments and methods of analyses. And so next, we'll take a look at some of the results of this research. The following figures were created for me by Dr. Banerjee, so I definitely want to give her credit for her work. And so when considering diversity, there are two common metrics of comparison. The first is alpha diversity, which represents the average species diversity of a single habitat. This method is also known as species richness, as it measures the number and types of species inhabiting a single area. And so beta diversity, on the other hand, measures the change in diversity of species between two different habitats. And so looking at this alpha diversity chart, we can see that bacteria is present in amniotic fluid regardless of chorioamnionitis infection, even in a relatively low count, indicating that the intrauterine fetal environment has an active bacteria, or not necessarily active, but has a bacterial community regardless of chorioamnionitis infection. I also wanted to highlight how chorioamnionitis modifies these bacterial environments. So in comparison to the control, which we can see here in red, we see modifications in the alpha diversity, for example, of the amniotic, the endometrial, and the placental environments. And jumping back in, we can see that there are more modifications in areas that are associated with chorioamnionitis infections, such as the endometrium, the placenta, the amniotic region, much more so than in regions not associated with this infection. So the oral, the rectum, the stool, etc. In the beta diversity chart, we can see differences between the vaginal and intrauterine microbiome. So we see here that the vaginal microbiome is represented by these multi-pointed stars, and we can see that they are pretty diverse. They form a pretty large cluster across here. I also wanted to point out another region for comparison. So we can take, for example, the oral bacteria, most of which is clustered here, but can also be seen in this kind of region in similarity to this bacteria here. And then we can compare that in comparison to, let's say, the placental bacteria. So the placental bacteria is actually almost as different from the vaginal environment as the oral environment is. Of course, there are some overlapping species and strains of bacteria, but this really does support the idea that the intrauterine space and the space in which the fetus develops has a unique environment as compared to the vaginal environment. And so here I'm going to discuss some of these findings in more detail. And so there are two theories that I wanted to highlight to give a bit more context to these findings. Again, we see the theory of the independent fetal microbiome posited by Dr. Agard's team, but also the defense theory of the placental microbiome, which was proposed by Kalpour et al. In 2014. This basically explains the existence of bacteria in the placenta as a mode of defense for diseases such as III. And so in terms of Coriomenitis, we can see that it is region-specific infection because within all three methods of inducing Coriomenitis, and we can see that it caused changes in placental tissue and the space in between these membranes and not in colon tissue, meaning that Coriomenitis is, again, region-specific. And also looking at alpha diversity data, we can see that LPS and IL-1 beta induced Coriomenitis also modified the placental and endometrial microbiomes, increasing the relative abundance of non-pathogenic organisms. And so this fits into Kalpour's innate defense theory, meaning that these modifications could be the fetal immune system attempting to defend itself from infection. And again, as I highlighted in the beta diversity results, the amnion has some bacteria that colonize it. It is not completely sterile. And Coriomenitis is not always associated with maternal urogenital infection. In this case, Coriomenitis was triggered with an intra-amniotic injection and progressed largely as expected, which supports the idea of the independent fetal microbiome once again. Finally, for conclusions, I want to discuss what this research means for the field of medicine as a whole. And firstly, I want to highlight that the human microbiome is still a relatively new field of exploration. And as Dr. Banerjee and Dr. Agard's research demonstrates, it's really a massive frontier for the investigation of human disease. For example, previous generations of scientists have not been able to explain the presence of urea plasma, a bacterial genus that is so heavily associated with Coriomenitis in otherwise healthy women. And so within the context of an independent fetal microbiome, we may further be able to understand this disease and why this bacteria is not always linked to this infection. In the future, the placental microbiome may actually become a consideration in maternal fetal medicine, the way that gastrointestinal microbiome is in gastroenterology. And so, for example, you may take probiotics or have heard of probiotics in yogurt for taking care of your gut microbiome. In the future, we may have similar treatments and a similar level of understanding for the placental microbiome. And so just to look at future projections and some ongoing questions, you may have noticed that I never mentioned that the bacteria in the intrauterine environment is alive or even where it comes from. And that's because we don't really yet know, not in Dr. Agard's lab, but in other labs in the United States. They are currently trying to culture this intrauterine bacteria and discover whether it is alive, what function it serves. And these are all really important questions to the understanding of the functional profile of this environment. We also still don't 100% know why this bacteria exists in the uterus. I mentioned one theory, the defense theory posited by Kalpour, but there are many others, and such as the idea that this bacteria exists in order to help prepare this environment for pregnancy. The bacteria might not even be alive at the same time that the fetus is in the uterus. And so there's much to know about this environment and about Coriomenitis infection as a whole, both at the microbiotic and pathological levels. And it really is exciting to learn more about this disease, but there's a lot to be known about Coriomenitis before its complications can really be prevented. I also wanted to elaborate a bit on specifically where I fit in this research. And so throughout my mentorship, one of the most common topics of discussion was the communication of scientific information. And so quite obviously, an immense amount of work and time goes into the infection of these critical yet common conditions within the laboratory. But this research is difficult to be understood, be utilized, or even be appreciated by the general public. And so scientists must really resist the urge to both oversimplify for catchy public appeal or overcomplicate their research to a level where the public can no longer understand it. And as I've continuously discussed, Coriomenitis is an infection that complicates thousands of pregnancies annually, but many mothers affected by it still don't have access to the research and resources that can actually help them. And so as I enter my academic and professional career, I hope to continue to educate others about science in a way that's meaningful and accessible. Finally, I want to give a massive acknowledgement to my teacher, Mrs. Nancy Bryant, as well as my mentors, Dr. Sohini Banerjee and Dr. Kirsty Agard, of course, without whom none of this would be possible. And thank you so much for coming to listen to my presentation today. I would love to field any questions you have about this project or about my mentorship as a whole. So I'm going to stop the recording now and